Thank God.
I'm really sorry what I said this one.
It's like an intimidating thing. It's complete first.
Hey everybody. All right, so basically, my name is Paulo Schwartz. I'm a student at the MSc BIG program, which is in the School of Social and Political Science. And this event is based around synthetic biology and the intersection between the engineering side and the life sciences side and the sociology and political side of which involves all the bioeconomy. So basically it's going to be very interdisciplinary, and we have really good speakers who know a lot about synthetic biology, and we're going to let them explain to us what is synthetic biology for those who don't really know it, and we're going to delve deep into the subject. And then at the end we're going to have a half hour for discussions and questions from the audience. So who's going to be conducting the interview? The meeting today is Pepe, who is the president of the Synthetic Biology Society, and he's got more about the event and introduce the speakers. Thank you for coming. I hope you have a nice event.
Hello everyone. So as Paulo introduced, we have a great panel today, and we're hoping to have a relaxed discussion about current issues in synthetic biology. And I'm the president of the Synthetic Biology Society, so Paulo and I just wanted to have a collaboration with students from the School of Biology and students from the School of Politics, because we don't really get to have events together and kind of see the whole different areas of expertise within synthetic biology. So thank you to all of our speakers for being here today. And as Paulo said, we'll have a few questions that we've set. We'll hopefully discuss them, and then we'll have some time at the end of the event for some questions from the public. So thank you all for coming today. So I'm going to quickly introduce the panel. If possible, they could just quickly speak in 5 minutes about the background and how they're related to synthetic biology and where their expertise comes from. So beginning from my left, we have Nick. He's a postdoc at Louise's Horsfall lab. So he's a researcher in synthetic biology. So you want to introduce your background?
Hello. So like Pepe said, my name is Nick Pantidos. I'm a postdoc at the Horsfall lab over at King's Buildings. My background is heavily influenced by synthetic biology, biotechnology, cell biology. So I'm a trained microbiologist from my undergrad. Went ahead and did a master's in biotechnology, and then I finished a PhD in cell and molecular biology. And a lot of my research was based on bioremediation, so essentially using bacteria to remove heavy metal ions from copper or silver contaminated byproducts or waste, and trying to convert it into useful materials to use for other processes such as catalysts or antimicrobials. And so now I've been working as a postdoc for the past 3 years and looking at bioremediation again and trying to do some metabolic engineering and just learning more about the pathways of nanoparticle synthesis from using bacteria.
Thank you. So we also have with us today.
I come at it from a different angle. If a research council wants to define synthetic biology, it's in order to make sure that the people who are applying for the grant are appropriate people to receive a grant and to deal with it in the right manner so they understand what they're doing and so on. And that happens in research councils. And well, at least in the beginning, when synthetic biology first began to be talked about, there was quite a tussle between EPSRC and BBSRC, both of whom wanted to own synthetic biology because it was the next big thing coming up. And BBSRC kind of won that, partly because they were willing to put more money into it. And after a while, the engineers began to think, hey, we're losing out here. This is an engineering discipline and we're not getting our fair crack of the whip. And you still hear that from engineers working in synthetic biology. And there's now a move led by Dick Kitney in London to rebrand it as engineering biology. So he's set up a journal called Engineering Biology, which is emphasising the engineer's side of the equation. And you know, actually, every time I look at a really top-notch synthetic biology presentation, there's a hell of a lot of chemistry in there, and nobody ever mentions chemistry. So it's a real amalgam of disciplines in a way that's very interesting. And I actually think you shouldn't try to define it too tightly when the new discipline is still evolving, because you will kind of repel people who might actually be able to contribute something really useful to learning. So it's not a great idea to try and focus too hard on the definition of the discipline. But the area I meet it most is in the regulatory sphere. So if you want to capture something in order to regulate it, you have to define it within the regulation. And people working in synthetic biology and gene editing are really concerned about having it classed as a GMO, because if you're working in Europe and it's a GMO, you have to go through the GMO regulatory system, and that's not fit for purpose. So a lot of what you'll have heard from scientists about definitions of synbio recently are desperately trying to say this is different from a GMO. And they have achieved that with gene editing because they've said there's no new genes inserted when it's gene editing. But they haven't achieved it for synthetic biology where you are actually putting new genes in. So, and an interesting thing from America, GM cattle in America, for regulatory purposes, are defined as drugs, which is an interesting category. It just won't work, but you'll find that out.
Thank you.
Complicated.
I agree, there's no single definition, and maybe we shouldn't try to put it in a box yet. So this is another big question, but from your expertise, what would you say is the true potential of synthetic biology in solving the world's greatest challenges from health, agriculture, or industry? Anyone?
I mean, in terms of genetic engineering itself, it has been used for decades now to solve everyday problems and health problems, food problems as well. In terms of synthetic biology itself, I think it's just an evolution of where genetic engineering started, basically. It's about, by throwing in the engineering, for example, discipline in there, it just makes it more, like we said, a standardized way to to increase throughput, essentially. And nowadays, with populations rising and we got more and more problems, we want efficiency, we want high throughput, we want reproducibility. So I think synthetic biology could bring a good toolkit to solve problems like that. So, for example, can be a good way to increase the throughput of science and going through to solve those problems.
So, I think synthetic biology can't be thought of in isolation. We've got so many things that are coming up now with synthetic biology. For example, big data being a massive issue at the moment. Nowadays, my lab, we don't. Generally, all the labs in my building, we don't really know what we're going to do with all the data we generate. Just trying to organise it into a fashion where we can remember in 5 years' time what we did this year is going to be an issue just because of the sheer amount and the categorisation. So that extra capacity to have data has really helped things like synthetic biology. The 1,000 Genomes project, I don't know if you guys are familiar with that sort of thing. The abundance of information we've got of different genes, so just, imagine how that helps the design of different tools. We have all these different alleles of different genes that could be used for genetic engineering. It's I think all of these things together are helping, achieve things that would be considered new and wonderful. I don't know, have you guys read, there was an article a couple of decades now ago, could a biologist build a radio? Have you ever read it? And one person's nodding his head. Now, forgive me if I'm going to quote this wrong, but the idea was basically, if you gave a biologist a radio and you tried to make him understand it, the first thing a biologist would do would smash it to pieces. Then he would basically look at all these pieces and go, aha, there are 400 pieces. He would then classify them into groups, which he thought. And then eventually he would start sticking two pieces together and saying, oh, I now understand this pathway. And he basically that, eventually after having stuck a few pieces together, he would now decide that he's decided how this radio works and that he's defined the process, and that was that. Of course, that's absurd when you think of it from an engineering perspective, but the reason that a biologist would do that is because if you think about that from an organism point of view, nothing is in general. You do the best you can do. Whereas nowadays, where we have all these things being categorized, you can start thinking about these things from an engineering perspective, just like an engineer would in the first place. And it's giving biologists the capacity to start actually really being able to build the radio and thinking about that from an organism point of view, and start to do things like building synthetic organelles, which is another paper that just came out the other day, and really manipulating nature in new and wonderful ways.
I think it's kind of limited by our imaginations. It could do so many things in so many different places. It just takes some really bold thinking about about what new things you could possibly do with this. There's the chap who sends around these, the Californian guy who runs conferences, it's in Biobeta, what's his name?
He was an Edinburgh student.
John.
Cumbers, yeah, John Cumbers. I get his blog regularly, and the blog I got a couple of days ago described how an American company has received $300 million of investment to produce artificial meat from extinct animals. And the company was described as a unicorn. So, you know, I mean, who's going to pay money that will cover those costs? I don't know. It's getting a bit like the dot-com bubble was with IT in the 1980s or so. And there's a lot of money beginning to be put into synthetic biology, in particular in America. Now, most of it will come to nothing, but they invest so much more than we do that they will probably get there first. But I do sometimes wish we'd be just a little more upmarket and imaginative and think more radically into the future. What could we do with this new technology? Because it's really, you know, and I agree about linking it up with big data and artificial intelligence and you know, digital sequence information. That's going to be enormously important in the future. And it's almost entirely AI. It's really important. It's really important to link these new digital technologies to the biology on which they're going to be working, and to get those links working well. And it's a bit unfortunate that the government, having set up all these synthetic biology centres about 5 years ago and set them going, completely changed its focus. So everything is robotics, AI, smart cities, but everything to do with what goes with the artificial intelligence technologies, not the biotechnologies. They've moved away altogether from the biotechnologies, and I think they need to be pushed back so that all these new artificial intelligence technologies will pay attention to the biology that we're going to be working on. I think that's going to be really important. But the sky's the limit, really.
Yeah. I was reading about that yesterday and I wasn't sure if it was an April Fool's joke from Bioeconomy.
Well, it might have been. I didn't think.
Because they were talking about this company called Mamuti that was a PhD student from George Church's lab that went off to set up a company to sell synthetic mammoth steaks.
Right, yeah.
But I think it was I don't know.
Well, it's imaginative.
Yeah, I believed it, so
Yeah.
it would make sense nowadays.
But also, if you looked further down that information email, you know, there was 500 million for this small SME setting up and 600 million for somebody else. There's an awful lot of money going into it. Just the same. Yeah, but
I wouldn't be surprised. So bringing this into our next question, many people are afraid that there's often a gap bringing innovation from the lab to the public and how to bridge this gap. So what would you say should be the balance of publicly funded research at universities and research driven by companies, and of course into that falls collaborations from research labs at universities and companies?
I would say there's not nearly enough coming from companies at the moment. There might not be enough coming from public research, but it would be really good if companies would begin to put money into it. I think that would be very useful. Why not? I think they're nervous about regulation. I think they don't know whether it's going to be something they could do in 5 years or 15 years. And 15 years is not an acceptable payback period for a company, so they're reluctant to step in there. If you look at the industrial biotech area, which is where the bioeconomy is going to be benefiting from synthetic biology, the companies who are currently incumbent in that area are generally big petrochemical companies. They're not going to turn themselves into fermenters overnight. You know, they're not going to replace their fracking site with large-scale fermenters. They're not going to do it. They couldn't do it. They don't know how to do it. It would cost them too much money. So they're not going to be the ones that pour money into Bio. If you look at the other big companies that might do it, I guess you might be thinking about the distillers and the brewers. They're pretty big, some of them, and they could set up parallel initiatives using synthetic biology to develop bioplastics, etc. But they're a bit nervous of the public reaction to it, and their food companies, well, whiskey is a food in some people's minds, and they're nervous about the public reaction to what they might do in the SynBio area, beginning to taint what they do with their current products. So they're not going to jump in with both feet. So you're left with a lot of SMEs who could do great things and who could set up whole new industry sectors doing things with synthetic biology, but they'll need government funding to get themselves across that gap. So I think there's a space there, but it probably still needs more public funding, because I don't think the big incumbent companies are going to start investing in SynBio to a large extent. And
do you think the UKRI and the agri-tech and sort of all these sort of bridging funding things are helpful enough? Or?
Well, they will make a difference.
Yeah.
Not nearly enough. Yeah.
This is a new, basically, the funding landscape has changed since I've been here over the last 5 to 10 years or so. And there are a lot more bridging situations now where you have the opportunity as an academic to find an industrial partner and look for funding, which can take you either from proof of concept to what they call mid-range processes, all the way through to technology level, depending on how mature your product is. So these things to bridge that sort of the gap between sort of finding something in the lab and having a product are coming. They're available now. The funding can be found, but actually, finding the right industrial partner, getting hold of that funding. There is still an emphasis, I think, from funding bodies that you want to have research which is new and exciting, and the problem is that once you've had the proof of principle, it's no longer really new and exciting, so you're kind of just going on the slog to try and get it to be a product. So to make that enticing and exciting is a difficult thing for an academic to even sort of think about it from a paradigm point of view. But I think more money is becoming available. I suppose it's a good thing.
I mean, from my limited experience in the field, I agree. I've been responsible, I suppose, for applying for a few of these funds that Alistair was talking about recently, where I haven't really seen them before, not as widely available anyway, like Pathfinder and BBSRC ones. And they do help a lot. They help get the research from the lab out into, or just push out the door essentially into either start a company or at least do some proof of concept studies. In terms of the big companies, I think money is an issue for them. They want to have the public's kind of support, but they are a business after all. They need to keep making money, and if they need to find a lot of money into a new type of research, then you better be high paying in the end type of thing. Whereas publicly funded research can be a lot more risky, and therefore the basic research a lot of that happens in labs, so in academia, for example. But I think collaboration is definitely key out there, because I've been a member of, I've been a part of a collaborative industry academia collaborative project, a number of them, and they work out very well. Everybody's happy in the end of the project, and more often than not, I see that by the time the project ends, the company is interesting to keep the project going, so they funnel more and more money into it, and everybody's happy in that respect, because the expertise stays comes from the academic side, and the industry is happy supporting that research in kind or with money, or if they have the expertise depending on the company. So I think collaboration is definitely good.
Who were these big companies that were investing?
In my case, I've been working with Diageo for a number of years now.
Whiskey.
Whiskey, yeah, exactly.
But working on their main product, which is the waste from the distillery,
Exactly.
not using distillation, brewing and distillation to produce something
Exactly.
new and different.
So, like for to quote one of their kind of figures, for every liter of whiskey they make, they produce like 10 liters of co-products, which
Yeah,
then is used, but they're also constantly looking for new ways to use the product to its best potential. You know, get
Yeah.
as much resources as
Yeah.
possible out of them before they can dispose it off, or you know, if they manage to do it very efficiently, then they don't need to dispose any of it off.
yeah,
And this, in my lab, we have a number of other companies that are working with us as well.
Yeah.
So it's, I think collaboration is definitely key.
but I think there's a bit of concern that if we really started to do that kind of thing on the scale that would compete with petrochemical-based products, and at a price that would compete with petrochemical-based products, then there isn't going to be enough waste material to fuel these industry sectors. And countries are beginning to run out of waste material already somewhere. So I think in Germany there is no such thing as waste material anymore. It's all used for something, and it becomes pricey. You know, it's not a free good to take away from the people you're relieving of it. They want money for it.
Thank you. How would you say that openness and intellectual property plays into collaborations with academia and industry, and if this affects your work in any way?
So IP essentially. I've had plenty of discussions about this actually with a few of a couple of companies that I'm involved with, and it's difficult to draw a line on who owns what sometimes, and the best way to avoid complications in the future is probably sit down and define who owns what before they start the project, before a contract is signed. Because after that things get a little complicated. But it just depends on the type of project you're doing. In biology, it's a little bit more complicated because of the nature of your product per se. Because it's a lot more often than not, it's a method rather than a product, unless you're, J. Keating and you're making artemisinin. But then again, you still be paid. Well, your IP would be the method. So it's a bit of a tricky subject on who owns what, but I think as long as things are just negotiated before the project, that should be avoided in the future.
So for us, I mean, we've also had issues with this. And it's, even so, before the contract, I'd say before you even think about the project, you need to nail these things down. We recently had an issue with a project which went through the funding round. It got funded by the funding body, and we need to sign a collaboration agreement with the company before it starts. And unfortunately, the collaboration agreement was quite complicated because it wasn't just between us and one company. It was between us and one company and another patent, another university which held a patent on one of the technologies that we were going to be using in that project. And the company and the university didn't want to talk to each other. And because that didn't happen, you don't get a collaboration agreement. You don't get a collaboration agreement, you can't start the project. You can't start the project. Basically, you just waste your time getting a grant application. So, I mean, luckily this all came right. Literally 2 days before the postdoc was supposed to start the project. But it was sort of a really interesting process to have gone through as a PR. Sort of, when you think about putting a project together, you think really about the science, and you think that everything else will just fall into place. And we do have in Edinburgh now, we've got Edinburgh Innovations and people involved who help with these things. But more and more it's becoming really, really important as a research level to think about these things yourself, so you don't run into these big problems, particularly when you're dealing with companies, which you will more and more now because of the nature of synthetic biology. Yeah.
But when synthetic biology was set up, there was almost a principled ethos around openness. Because the people who moved first with synthetic biology in MIT, came from the engineering and IT disciplines there. They had this notion that open source software was a good thing and proprietary software was a bad thing, and they wanted to transfer that ethos into synthetic biology. And there's been a lot of talk about almost the virtue of openness as an ideal, and the nasty real world commercial stuff that comes in when you just have to have the IP in order to do anything. You know.
It's a bit of a shock to the system, actually.
Yeah.
As soon as you go
Yeah.
from open up, Golden Gate system where you can basically just share plasmids left, right, and centre, and
Yeah.
everyone knows what everyone is doing, and you get that one gene which is really important, and suddenly
Yes.
sort of done. You don't see it coming necessarily.
Yeah. Yeah, and you kind of can get away with it in AI because you can develop things so rapidly. You're not going to hit any regulatory roadblocks. It's not, proof of concept is not nearly so complex as it is in biology. So you can just do your IT kind of thing and get it up and ready and get it out there on the market, and if people like it, you're a billionaire before 2 years have passed, and if they don't like it, you're out of business. Whereas with biology, it's a long time to get to the point where you've got your proof of concept, and then you hopefully, before you get to that point, be thinking about how it's going to be regulated, and depending on how it's going to be regulated, it's maybe 3 years, maybe 15 years. If you're in Europe, you'll never get on the market in Europe, so don't even try. If it's America, you could probably get there in 5 years, and that's why a lot of the SynBio stuff is happening in America and not elsewhere, because they have decided they are not going to impose very much regulation on gene-edited products anyway. SynBio probably still be regulated a bit in America, but not nearly so much as Europe. So that's one of the things that drags all of that into America and makes things happen faster in America. And so, presumably the openness thing might not be such a problem in America if you can develop it fast, and it's just one of those trade-offs you have to make.
I think they're generally faster because the patent
Well, they do patent
stories
it
is
as
that
well. Yes.
there's much more push in
Yeah.
the States to actually have patents in labs
Yeah.
and things like that. And I think because that is already set up, the capacity to even communicate between groups is much smoother.
Yeah.
It's still quite a foreign thing to most UK labs.
Thank you. So moving on to regulation, what, in your opinion, what do you think should be the best governance strategy around synthetic biology? You've mentioned the situation is very different in Europe as in the United States or other parts of the world, but what would you think is the best way forward to maximize the benefits of the applications of synthetic biology but minimize the risks?
Well, I could go first, but you guys should tell me what your experience has been with regulation first. Have you had experience with that? Have you thought about it much?
Not so much myself, it's mainly just knowing what you can and cannot use, because depending on if you want to commercialize it in the future, then you know not to use certain methods because they're either patented or owned by someone. So before you set off, you need to think of these things.
Is that ever an insurmountable blockage on doing something?
It depends on the process. In my case, I've been fortunate enough to avoid these roadblocks, but generally, like if you want to do gene deletions, for example, using CRISPR, that is now, it's owned by someone. So if you're using that method, you then have to kind of acknowledge them in a way, and then whether they want to license into you, I don't know how that process works, but if you want to avoid those issues, then you have to think of another way of doing it, which might present different problems because it adds time to the process. But that's probably as far as my experience goes.
For plant synthetic biology, I suppose it's where you're going to plant your synthetic biology crops. Scotland's got a no GMO regulation at the moment, but England doesn't. So you can grow your GM wheat. One of the first GM wheat trials was done in Rothamsted. I think it's into its second year now, actually. That's really exciting. But you wouldn't be able to do that here. And I was talking to Joyce about whether Wales would be an issue as well. So, but I think Scotland is probably the worst of the United Kingdoms at the moment. You wouldn't be able to do it in Europe because of the European something much stricter rules on GMO. Whereas in the States, obviously, you'd be much easier going about doing that. So yeah, where you would, who would you collaborate with, and what companies you would have to get together with would be quite a big decision, I suppose. For that.
And one of the things about regulation is that if the regulatory system is really tough and expensive, then if you're an SME, you could never do it on your own. It's retail market at the end of the day. You always have to end up doing it through a company, and that means you tailor what your research is going to do to a notion of which company will like what you're doing and therefore might buy you up at the end of the day. And that means that if you wanted to do something really radical that would compete with the current strategies of the multinationals, then you wouldn't go there if you're sensible. Probably means you wouldn't do anything that wasn't on a big commodity crop, because there aren't many big companies working on non-commodity crops. Rice, wheat, cotton, soya, obviously grape. These are the five. And if it's not working for one of them, if it's going to do something really great for apple growers, then you don't have much of a hope of getting that through the regulatory system and onto market the way it is in Europe at the moment, whereas you do in America. In America, there are already some pretty daft applications, actually. The Arctic apple is on the market in America, having not been regulated at all because it's done through gene editing. And it's Okanagan Specialty Fruits is the company that does it. And it doesn't go brown when you cut it. So the idea is that you can use it in all these pre-prepared salads that you get in supermarkets. You don't see apples in these pre-prepared salads because they would go brown. So if they don't go brown when you expose them to the air, you could use them for that. And they'd be cheaper than, say, pineapple. And apparently the only reason people use pineapple is it doesn't go brown. And so on. Or because it sells them as well, I guess. But, they're weighing up all these kinds of things. And that got through onto market, I think, in less than 5 years. So the development costs were supportable. But America still does some pretty daft things like treating GM animals as if they were drugs. That's not very sensible. Apparently the GM salmon in America, it's not completely free of public influence on these technologies, because I think the public influence has to do with the way they're regulating GM animals. And the GM salmon is actually produced in Canada and Chile. It was approved for human consumption in America by the FDA, but they didn't approve its being used on American fish farms. So it's produced in Chile and Canada and then imported into America. And I think that was probably due to some public pressures from Californians who definitely don't like GM stuff. There's a lot of anti-GM in California. It's nowhere is it great. People say Canada has got the best regulatory system.
Canada's amazing.
I think it's okay at the moment. And it's all about how you capture something new for your regulatory system. And the Canadians do it by saying, is this new to Canada? Is this something that's never been grown in Canada before? And if it's new to Canada, it then has to go through the regulatory system. What that means is even conventionally bred crops, if they're new to Canada, have to go through the same regulatory system. Now, that's not too bad because they're quite sensible about not putting ridiculous constraints on things that are clearly perfectly safe. So it's quite easy still to get through the Canadian regulatory system.
Do they actually have the term genetic modification in Canada? I think I've heard that there was a paradigm where they called something else. So even that sort of negative connotation.
It could be a recent thing. I mean, it was called GM originally. Yeah.
Just think that apparently the Canadians, as customers, they just don't seem to worry about this as much as the American counterparts. It's just interesting to see different populations doing.
Well, they're making so much money out of it.
Yeah.
Yeah, I mean, quite a lot of their agriculture.
But there's a trust that's been established that seems to be in place.
Yeah.
Sort of.
Yeah. So if you want to produce something, Canada's your place. Yeah. And then if we want a new regulatory system here, maybe think about Canada as a model. Yeah.
So you were mentioning this, the opinions of the public in regards to something that we as scientists might completely know that it's completely and utterly safe. For example, I'm from Spain, so in Spain, you can't grow GM crops except BT corn. I guess because it's been grown in so many places that they knew for sure that it was safe and they needed to grow corn. So they made an exception in the law to allow BT corn, but not any other GM crop, and it's not up to discussion. So would you say that there's more that should be done in terms of outreach from the scientific community to policymakers or the public in terms of the applications that are on the less risky side but have been used?
There's an awful lot of nonsense talked about the risks, actually. You know, there's no evidence at all of any risks having arisen from the GM crops that have been grown up until now. Synthetic biology, gene editing, if anything, are likely to be safer because they're more targeted and we can tell more clearly what we're doing with them. The one kind of caveat, I think, is gene drives. I do think if it's a gene drive, we don't actually know enough about that yet to know that we couldn't permanently alter an ecosystem using a gene drive, so that would be bad news. I think what's going to be difficult to distinguish in the public is distinguishing gene drives from the more conventional applications of gene editing, because they both use CRISPR-Cas9. And I was at a meeting, I gave a talk at a meeting of the Scottish National Party's annual political meeting on GM crops. And there was an NGO representative at that meeting who spoke about gene editing as if everything that was gene edited was a gene drive. And so he really reinforced the anti-gene editing perception in his audience by claiming that, you know, these are all the negative impacts you will get from gene editing, and he was talking about gene drive all the time. So that, I think, is a difficult one, and if you ever get a chance to speak to the public about CRISPR-Cas9, please make it very clear that there are things called gene drives, which we're going to be very careful with, and there are things called gene editing, which we can have much more confidence that they're going to be safe.
Yeah, I don't have much to add to that, actually. I think, the conversation is ongoing. We've made great strides, I think, in the last few years in terms of convincing. I mean, this is done well in terms of synthetic biology having a positive spin and people taking it on board. There was I think the Telegraph, which is an interesting place for it to be, there was a study which showed that I think most people under the age of 30 or 40 are now much more, they would be happy to eat GM food. They don't see any issues with it. I'm not too sure whether it was just the UK or Europe as well, but it basically just goes to show that the paradigm of GM food being a bad thing and GM products being dangerous is changing. I don't know. It's difficult to actually say just how bad the Europeans' decision was for not allowing synthetic biology to go ahead. That's going to take another 5 to 10 years to probably change again.
But actually, if you look at it was the Council for Justice of the European Union that made this decision, and it looked at how GMOs are defined in European legislation, and it looked at what synthetic biology, so what gene editing, it was only about gene editing, it looked at what gene editing does. And if you look at the argument they put forward, it's legally very rational. You know, if you're only thinking about the law, then that was a reasonable decision to make, that gene editing is the same as GMOs, because they have similar kinds of impacts on what the organism is at the end of the day and what it can do. And I don't know all the legal jargon, but it certainly was enough to convince me, and I asked a colleague who's a lawyer if that was a valid take, and he said, yes, absolutely, but it's horrendous for the technology. And so they just make their decisions based on the law. And it was taken to the European Court by the French government based on an initiative by a French NGO. So it was an NGO-formulated attempt to make sure that gene editing was captured by the EU GM regulatory system. It's actually caused so much angst and annoyance in the scientific community in Europe that I can't help thinking they might just start to begin to change their minds.
If you look at food security as a major driving priority of both the UK and Horizon 2020, all your EU funding, one of the reasons being, if you look at predictive models for climate change, there was a paper a couple of years ago basically that the rate at which the climate is changing in terms of temperature increase is going faster than basically a lot of plant species can evolve, particularly grasses. So the actual ecosystems for grasses in 2050 and 75 and so forth will be vastly changed by the changing climate that we expect to occur. Our grass ecosystems include things like wheat and rice and all of our sort of major staple crops. So while you'll be having places in the States which will be able to introduce new varieties by using breeding strategies, as well as things like gene editing to make things that are going to cope better in those environments. So all of your breeding programs are going to be relying on gene plasm, which doesn't have the capacity potentially to deal with these climates, which is going to be a major food security issue for them in the future if they're not careful.
And also crops are very region specific. So when Monsanto bought the Unilever plant breeding facility down near England, what they wanted was to get their hands on the European wheat varieties, because they couldn't develop crops for Europe by modifying American wheat varieties, because the American wheat varieties don't grow well in Europe, so they needed the European varieties. And so for a lot of these areas, it's not as if we'll be able to rely on developing them and we'll be able to buy some and grow them in Europe if we want to. We just won't have the crops that are tailored to grow in the European climate.
I saw it in Zambia with soybean varieties from the States, which were massive, the biggest soybean yields you can get. I'm trying to grow these in the Zambian field, and they're like this high, compared to the sort of things that are found in Nigeria, because Zambia is trying to get a soybean crop going.
Yeah.
So just, when you've got things which have been established for 50 years now as being massive producers, take them to different parts of the world, and they just don't cope.
One of the encouraging things about Africa is they've got their own scientists in place, they've got their own regulations in place, and they're starting to modify their own indigenous crops with using GM.
Things like cassava is really exciting, actually.
Yeah, and so they're beginning to develop crops made by Africans for Africans, and they're not going to bother about Europe anymore. What was keeping them back over the last 15 years has been that they sell crops to Europe. And I think they've probably now got markets in places like China and India that means they don't need Europe, and they're going to go their own way. So Europe's going to be left on its own out there.
So talking about innovation, as you were mentioning before, how the government specifically has been relating to synthetic biology. We had the UK Synthetic Biology Roadmap in 2012. 5 genome foundries opened up across the UK. So what do you think is the place of the UK in synthetic biology within Europe or within the world?
Take a step back from that and just look at the investments in the gene foundries. I'd say one of the first things that we should be doing is trying to use them effectively. There's a classic situation with funding in the UK, and maybe I'm being cynical, even if he knows it. We tend to do that. For example, this happened with deep sequencing, and that came out about, 10 years ago now. UK invested a whole bunch of money in getting hubs around the country. So the North Hub, the East Hub, the West Hub, the South Hub, where a whole bunch of aluminum machines were brought up and bought to these specific places. And they had a whole bunch of scientists come in to run these things, which is fantastic. That they get administrative and business staff, not so much. And what you're trying to do is build facilities here where people will come and use you for deep sequencing. Because they had no business program attached to getting the funding, they were supposed to be self-sustainable after 5 years. A lot of them fell flat. And then they were reinvented again. We have Edinburgh Genomics now, but that's actually a second generation deep sequencing facility. So what I'm hoping isn't going to happen with the gene foundries is that we end up with better business models associated with them. And I think Edinburgh's doing pretty well. It's being a bit slow to take off, but it's doing better than I think some other gene foundries. But yeah, I think they are state of the art, and the things you can do with them is amazing. The capacity is interesting because you have to ask much bigger scientific questions. So one of the things the gene foundry in Edinburgh does, instead of making one plasmid to do your experiment, you need to make 96 plasmids to use a whole nice plate. You wouldn't go to the gene foundry just to make one construct. You'd need to make 100, which for £3,000 for the plate. So immediately you need to start thinking much, much bigger in terms of your experimental strategy, what questions you're asking. So I think to take advantage of all these new tools is going to have to be a changing of how we design our experiments, and that might also change in terms of funding from research councils. And I think getting those things all connected is a big challenge at the moment. Yeah. But we certainly have the capacity to compete internationally.
Yeah, I think it's the technology jump there is quite big, and I think it's Alistair's argument that's causing the bottleneck here. That experimental design needs to change, because I've been to the Genome Foundry. There's the capacity they have to do constructs is tremendous. I mean, you're going from something that you can do in the lab that takes you 2 weeks and you design one plasmid to in 1 week you can design about over 1,000 different constructs, basically, right, for half the time. So it's about training people with that mindset. It's about getting the word. It just spread the word that these facilities actually exist and they're there to increase your productivity in the lab. I think people know about them, but they're not actually sure how they work and that they're not as hard to actually start using them. It's not, you don't have to change everything you do in the lab. You just have to go and speak to the facilities and see how to do it, and ask them how to design experiments or design experiments together, because it's not, you don't have to change everything. You just have to conceptually change what you're doing rather than like the direction of your project essentially. And you can have a lot more data. Hopefully enough data you can handle and not too much of it, but that's another discussion later.
I don't know much about this, but I do have a question. What can we do in Edinburgh that Twist can't do cheaper, or Ginkgo?
That's a good question. So Twist and Ginkgo make sequences. I'm not too sure how well they assemble them into different constructs, and maybe they could do that cheaper as well. But in terms of, I think the idea would be that the foundry doesn't actually make its own short pieces of gene fragments. They would order them in from Twist or Ginkgo in large quantities, and then you could build up your different mixtures of that. So yeah, that's whether the gene foundry becomes relevant, because maybe Twist decides to buy its own gene foundry as a capital investment. And basically you just need to go to Twist with your experimental plan.
I'm not sure how Twist and the likes of Ginkgo actually how automated their processes are. The Genome Foundry itself, compared to the other foundries in the UK at least, is a lot more integrated. So you can get from concept from design to a product straight, fully automated, pretty much. Whereas in other foundries, you do one bit automated, then you physically have to do a little bit of manipulation, manual handling, and then take it to the next automated bit. I think the Edinburgh Foundry is the most integrated one. But in terms of yeah, they make large constructs. So the Yeast 2.0 project, I don't know if people are aware of it, where they're assembling a fully synthesized yeast chromosome, essentially, yeast genome, and there's like 11 different chromosomes in it. And it's a huge, like, so many different partners are involved, and the places like the foundries basically are heavily involved in these, because you need to assemble this huge like hundreds of kb size base pair size fragments together, and what the foundries do is, yeah, they order in smaller pieces of, say, primers or oligos essentially, and the foundries are used to assemble them into large constructs. But that can only be done efficiently using automation. Like, if you sit there with your pipette in a lab, you'll be there. You know, you'll be 80 years old still working away doing half the chromosome. So it's automation is definitely one of the future in terms of productivity.
I think there's competition there. So I don't know if anyone uses SnapGene. Anyone know what SnapGene is? Basically, it's a plasmid assembly tool. It's really one of the coolest things that you find on the internet if you're a biologist, a molecular biologist. Basically, you can just build your own vector, and it's all colourful and sort of very user-friendly. One of the drop-down menus now has, it's called Plasmid Design or Lab Design. It's basically a link to a group which does vector assembly, just like the Genome Foundry. But they're based in, I think, in China. And they've got a couple of hundred people working for them, which are basically business-orientated. So they will take the 20 vectors that you design in SnapGene. You basically just click on that button in your program, and it immediately gets sent to the ordering website, and bang, then you can order it, and then hopefully you'll get them sent to you in a couple of weeks' time. Rapidly going out and collecting all the known parts of different kits that people have, all different Lego pieces, let's say. They're not very good at it yet for plants, but they've got reps all over the place now, looking for people to get interested in it. So it'll be interesting to see how much having a local gene foundry versus having something at a distance will actually impact on research decisions and strategies.
Are you stuck with your current business model, or could you change it?
What the Genome Foundry?
Yeah.
Yeah.
Yeah, when I was in IGEM 2 years ago, we did a systematic review and we had some interviews with the heads of the Genome Foundries. The 5 of them, and Edinburgh one was the only one that was like fully automated and seemed to be going in the right direction.
Yeah.
But yes, as you guys were saying, there's been massive changes in technology in terms of big data, AI, automation. Now I think Opentronics is the new startup company. You can buy like a lab robot for £5,000. Like how much like that would have been, or how that becomes feasible now for like even smaller groups to just have automation at a smaller scale. Or as you were mentioning, I think I've also heard of other companies that you could just order them to do whatever set of experiments and then they just do it for you. So what do you think the future perspectives look like? I know we've touched on a lot of these topics, but if you would be more optimistic, what would you want to see that has been accomplished in, let's say, 20 years' time in synthetic biology? In the perfect world case scenario.
I think having full global standardization would be a big step. I think there's sort of this general push that people think synthetic biology is going to replace wet lab work. It'll replace some of it, but probably the boring stuff. A lot of time that students spend when they come into the lab is learning basic cloning techniques and sort of things that actually some are really good at and others aren't. But they'll both be equally good at, say, experimental design. And I think things like standardization and having these tools given to other external companies will speed up the questions you can ask and the experiments that you can design. I think, yeah, that's the best I can hope for from a rather general scientific perspective.
It's hard. Excuse me. It's hard to pick one standard because there's so many pros and cons on each one. So I think there's been loads and loads of discussions on this because you have like the way biobricks work, the way you have a Golden Gate assembly, Gibson assembly. Then the Americans in the US, they have another system. I can't remember how it's called now. Basically, it's just how do you choose which one to use, and how do you convince every lab in the world to stick to that standardized system when you have so many different pros and cons, and new ones come up, every year. And so if you choose one today, who's to say that something that comes out next year and you're like, it trumps everything else, but all the regulations are based on that one standardized system that you chose last year. So it's I completely agree with Alistair about I think a standardized system.
All standardized systems still evolve, right?
Yeah.
So I think it's an interesting question. I think it may be that we have a variety of different systems out there, keep going in different directions, and people choose horses for courses.
Yeah.
Maybe the knowledge will be about knowing what different systems exist, or maybe that'll be down to the actual company. You wouldn't have to even think about it eventually. But yeah.
So synthetic biology is a kind of platform technology. So I think it's going to evolve by becoming more and more integrally combined with artificial intelligence and perhaps robotics as well, and so on. So I think it will depend partly on where these technologies go, and it's a bit difficult to predict. But I think, as I said earlier, the future's really there for you guys to invent, in a sense, and it's the capabilities of your imaginative thinking that will determine where it goes at the end of the day, with the caveat that hopefully we're going to get our regulatory systems into some kind of order and make them actually fit for purpose so that they can keep things safe but not stop innovation from happening. That's one of the things I've been one of the committees that I'm on, and we've kind of got a UK government initiative in place where they've set up a few committees that have actually got the remit of making sure that our regulatory systems are proportionate and adaptive to the needs of innovative technologies. So that's there now in the UK government. And I was in London last week at an OECD, and there were people in Canada and the Netherlands both talking about having a similar approach there. So if that kind of thing catches on and we begin to change the instructions that governments give to regulators, not keep this safe at all costs, but keep this safe but don't destroy innovation while you're doing it. And that will be a different kind of pressure on regulators. At the moment they only have pressure to be risk averse, because they might get sued if something goes wrong. And I think they're going to see a lot of pressure in the other direction. So that might make that much difference to the way it's regulated and might allow a lot more things to happen than otherwise would.
Well, thank you for all your answers to all of my many questions. That's all that we had prepared for the discussion. As we said, we have about half an hour now for any type of questions from the public to any of our experts or all of them at the same time. So I hope you can maybe touch on topics that we haven't touched on, or if you want anything that should be clarified, or just really any question. But first we could just give them a round of applause.
Maybe a bit more on the standardization. What do you think are aspects where the design of the experiments could be standardized more?
So I was actually just thinking about what you were saying about standardization of different parts. I mean, the great thing about DNA is that it is standardized. We'll probably get to a point where you can use either Moclo or biobricks or whatnot, but basically these little ends that you put onto your piece of DNA that's of interest, it probably will be a problem that's not very difficult to solve at the end of the day. The other things like contigs, when you're looking at genomic maps and things like that, can you apply the same standards to different organisms? And I think that's coming out now with things like the 1,000 projects and things like that. Did you have a more specific question though in terms of what you were thinking?
Like, I personally am mostly looking into metabolic models.
Complex models.
Yeah, so like, for example, there is maybe three standards. Like one most popular is, it's most popular because it has most functions and most people use it.
Is this looking at genome scale models or like particular pathways?
Genome scale, yeah.
Yeah, okay.
So, but I'm sure that it's gonna evolve definitely. I think the question is like, into which direction? So yeah, like that was just the.
I'm only just getting into genome scale models. I'm actually really interested in them. But that's the fact that you've got different standardization is really interesting. I didn't realize that was an issue. Well, of course it would be. Yeah, I mean, do you think that you could have a standardized agreement on one, or do you think there's strengths between them? Or could they be combined?
Like currently, it's more like that. Each year, they manage to implement different aspect of real metabolism into the models. So I would say that it depends, like when there would be kind of the tipping point where new things would not be discovered as fastly. Like from what I've read, it seems that it's still just on the kind of on the acceleration path, right? So it's gonna take a few years.
And are those kinds of standards a bit like regulations? If you impose a standard too early in the development of the science, you can stop certain kinds of science from developing in the future.
It's actually interesting because in that case, mostly software.
Yeah.
So the standards are really just how things communicate. So of course you can always walk around it, but it takes a lot of time. So it's better to just stick to one thing.
So if you're looking at for a communication system, that standard will limit the kind of communication system you can have, and somebody might come up with a fantastic, much better communication system, but because it wouldn't fit the standard, it would never be adopted. Is that a possibility?
Yes.
Yeah.
It's hard to know. Like I'm not familiar with genome scale modeling. It's out of my depth on that one. But I imagine it's quite a nascent field, quite new. And so, no, is it? I'm not sure. But if that is the case, I don't know if it's too early to tell which model would be the best. I don't know if it needs to be supported by real lab work evidence to then make a more informed decision on which one to go for. So I think knowing a little bit of have some scientific kind of wet lab evidence to support your model is definitely a good thing to have.
Are these like the underlying algorithms that look at the flux between like multi-intersecting pathways and how things get chosen, or how you predict where a particular thing will go? Yeah, I can imagine that must be a nightmare. Yeah, so I think the best thing we can do is start like really understanding characterizing the kinetic properties of specific enzymes in systems. So one of the issues that lots of these things have is that you very seldomly have one organism where all the proteins in that organism are actually characterized. It'll, for example, a protein that phosphorylates glucose. You won't have it done for the organism that you're looking at. It'll have been done, like if I'm looking at Arabidopsis, which is my model plant species, a lot of these enzymes that we look at have been done in tobacco. And nobody's bothered to do them for Arabidopsis. You can't ask a funding agency to do that because that's boring. So you'd have to get a PhD student to do it, or something like that. So basically, developing a genome scale model for a single organism where all the different proteins are characterized, that would be pretty cool. And I don't know if that would help, but I'm assuming it would get you in the right direction in terms of predictive power. Yeah.
Who's looking after the standard development at the moment? Who's going to maintain it or
I don't know.
own it at the end of the day?
As in well, the kind of the framework or the software that's most popular now is mostly curated by community people who use it.
It's not very stable or robust. Yeah. British Standards Institution was interested in doing that kind of thing. I think they had a project going with Dick Kitney on doing something like that, but I think they fell out. And I think Dick Kitney's now working with the National Physical Laboratory. That's probably a different kind of standard.
I was actually wondering if there are any reasons that have come to your attention, like cultural or historical, why some countries are much more open to innovation while Europe is holding back so much.
I'm not well positioned to say because I don't have a.
It's very much outside the field of biology.
Yeah.
Is it, who's a European here who has a feel for this? Does anyone come from?
You mean we're not?
No, but I grew up in South Africa, so I don't really have a feeling for what the European mindset is with regards to this. I sort of appreciate it, but I think I'd be.
I don't think Europe is anti-innovation. It's doing an awful lot of great things in astronomy, physics, robotics. So it's just this food-related biotechnologies that Europe's got a problem with. And I see it as mainly politics. So you've got similar pressures in America for stem cell therapies and strong reaction against human embryonic stem cells in America. And whereas we're not too bothered about that in Europe. And that's because American governments listen to the religious right, focus groups and publics. In Europe, our governments listen to the environmental groups. And the reason they listen to them so hard is because several European governments are coalition governments that rely on the Green Party to get their legislation through their parliament. So they won't do anything that will annoy the Green Party. And that's really what's behind it, more than anything else. I mean, pressure groups get pretty fed up with piling on the pressure if they're not having any traction with doing it. And they'll move somewhere else and see if they can get some. So they had such spectacular success with GM crops that they really got kind of overwhelmed by their success, and they've now boxed themselves into a corner where they don't see a way of changing their minds. It's a bit like us with Brexit, you know. We can't work out how we really want to change our minds, but we can't work out how we possibly could. It's a difficult political situation to be in, and I think that's got a lot to do with it. It's not so much what the average person believes. If you look at the people who say they don't like GM food or GM crops, there are only about 15% who say they wouldn't under any circumstances eat GM food. Even if it was better for the environment, they wouldn't eat it because it's GM. And that implies that these guys are ideological in their beliefs about GM crops, and they're not going to change their minds. It's like having a religion. Just talking to somebody about how it might be otherwise is not going to make you change your mind about your religion. And it's like that. Where most of the population who are kind of saying, yeah, well, we hear a lot of negative things about it, it might not be so good, I'll maybe tick the no box. These people are going to change their minds if you give them good information, better information, more positive information about the technology. And of course that information has to be valid and evidence-based, but otherwise you end up in the same camp as the NGOs, so you do have to stick to what's scientifically factual. But it's very possible to change people's minds if they're not in that ideological group. And if they're in the ideological group, then you can forget about it. Just try and stop them having too much influence on anybody else.
I was going to ask, mainly on applying SynBio on healthcare and medicine. So I've been to one discussion recently about antibiotics and the kind of thing where if they ran out, are we just all going to die? And the question is, I was reading an article about prospects on the field, and there's not that much talk about CRISPR related antimicrobials and that kind of things. And I was gonna ask you, what is the reason for that? So whether it's just restrictions or too strict regulation, or is it just not enough research input from companies?
We kind of touched upon it earlier, I think. So basically, I think patent restrictions definitely a big thing because, I mean, there was a big argument on who owns it a few years ago. I think they just settled it now. But I'm not, don't quote me on that, but there definitely was the argument of who owns it. So if you ask who makes these antibiotics, it'll be, you know, the big companies, big pharma companies, who invest millions and millions on creating new antibiotics. And lately, that hasn't been going. They haven't been funneling that much money into it because it's just not economically efficient for them. So that means that if they employ things like CRISPR, that proves another obstacle for them to make money. They are a business after all. They need to make money to survive. So they want to develop. They want to have their own tools. Basically, they want to have that freedom to do that without restrictions on on patents, regulations like that.
Did you mean CRISPR in terms of generating new antibiotics or in terms of modifying organisms so that they
So
are more...
one idea would be to use them as a means of attacking the microbes, so a very specific means of attacking specific microbes. So essentially using the CRISPR as a drug, essentially, instead of making new antibiotics by using CRISPR.
From what I've read, CRISPR is not as precise a cutting device as it should be for it to be used as an antimicrobial, I think, because there's a lot of literature being published every day basically on, trying to make it more precise, trying to make it to not cut everywhere in the genome or only bind to your specific target. If you employ something like that on living organisms, on humans, for example, you can imagine what's going to happen if you start binding to other places. That can be regulated, right? I mean, easily, especially because you would only be using it for, say, bacterial infections. But I don't think there's enough research that has gone into it to be used as an antimicrobial currently. But there's definitely the potential to be.
I think in terms of the patenting now, it's... Yeah, maybe someone else can correct me if I'm wrong. As far as I know, the resolution of the patent was for Cas9 only, so not all the other Cases, and it was only for eukaryotes, potentially, I think, as well.
I think the Broad Institute got the patent for eukaryotes, and Jennifer Doudna in California got it for her original application,
Which was
bacteria.
Bacteria,
So they actually
covered everything.
Because she originally published it in bacteria, but didn't actually patent it in mammalian cells. So then George Church and the Broad Institute patented it in that, and they actually split the applications.
Right, but that is still just for Cas9 and not for the...
Yeah, only for Cas9.
Yeah, so it's interesting, all this patent war went on for 10 years.
And
in
it's
the
only
meantime, another 5 Cases came up, and now we've got another 10 in the last 1 or 2 years. So it's been an interesting sort of ideological campaign in terms of lawyers getting rich, ultimately. But yeah, you can imagine, I think companies are starting to make their own tailored Cases for particular applications, and that might be the next thing, in terms of efficacy. But yeah, I don't know. We might see something come to market in a few years' time, but it's still really, I mean, literally new Cases are just being discovered at the moment. People are obviously steering away from Cas9. It's mainly just a research tool at the moment. Because one of the reasons it's a good research tool is because it has been established. And bear in mind that Cas9 itself is only about 5 years old. So it's really, really brand new in terms of the questions that we're asking. It's being able to engineer plants, for example. It takes a couple of years to get these things up and running in the lab anyway, even when you have Cas9. So, yeah, I think various fields have moved faster, but it may take a little while for things to come to market. I don't believe they aren't doing it though.
In terms of antimicrobial resistance, I think one of the things that's increasingly talked about is bacteriophage technology.
Yeah.
And I guess, synthetic biology there could actually do some quite useful stuff, I imagine, but I'm not really knowledgeable enough to know if that's the case or not.
I think I would also add, there's some groups working in CRISPR as antimicrobials, but one of the main disadvantages is how you deliver it, like the delivery of the CRISPR-Cas system, because you wouldn't want to put it in a plasmid and express it. So if you want to deliver it as a protein, it's hard to get it. So developing new viral vectors or like phage mix and stuff, I think is one of the main limitations too.
Viral vectors is something I'm interested in because that's something that it would be quite easy to scare the public about. And I think there's an education task to be done there to get people talking about good viruses and bad viruses. They now accept that there are good bacteria and bad bacteria. I think they need to get a strong message that there are good viruses and bad viruses, just the same as there are with bacteria, and kind of preempt any attempt to have a campaign on trying to ban the use of viruses in synthetic biology.
One of my lecturers was saying that she saw this piece of news in the article in America, and they were talking about an outbreak of whatever bacterium, but they kept calling it a virus. So she wrote, it's like, this is a bacterium, not a virus. They're like, we know, but a virus sounds more scary.
Yeah, that's what they do all the time. Can't win.
Yeah, perhaps it's like understanding that even like a phage, a viral vector could be just the proteins of the capsules, not actually any viral gene inside, and maybe a virus is just a piece of DNA proteins.
I think some of the successes from gene therapy programs will probably change public minds. Like, for example, Cystic Fibrosis Trust in Oxford, they spent many years trying to deliver plasmids to people's lungs. Obviously, lungs are a major issue with cystic fibrosis patients. So they developed all these kind of oils that you can encapsulate the plasmid in, and sort of shuttle it into people's throats so they can sort of spray the lungs full of this sort of oily substance, and then the plasmids could soak in. Efficiency is really low. But obviously everything shifting to viral vectors now, it's just the delivery system is so much better. And I expect, all sort of things like muscular dystrophy and all these which are treatable by gene therapies. You should see some successes coming out sooner or later. That could really change public perception.
Which would you say would be the next successful clusters or regions working with synthetic biology in like 15, 20 years?
What do you count as a success?
Places where people want to go work for.
Oh, right.
Is it going to be like the US or other places? Edinburgh?
I think the UK is doing pretty well for itself in an EU perspective, particularly England, in terms of its current positive drive towards synthetic biology and its investment in the field. And the US is always going to lead the pack, I suppose, just because they're good at funding things like that. Politics be damned. But I think, Australia is also trying to invest money in synthetic biology. They never actually have enough funding to really do as much as they could, but there's some really good synthetic biology going on from plant sciences in Canberra. I suppose you could think of China as well being up and coming, and there's a massive increase in good research coming out of China in the last 10 years. They're really making a big push to bring expertise into China and send students out to get that expertise and bring it back. So I think, globally, US, hopefully us, with China as well, probably the big boys. That's not unexpected, is it?
It might begin to differentiate that if you want to do Symbiome health, then there will be the go-to place. Symbiome plants, microorganisms, that they won't necessarily be happening in the same places. I think we're putting a lot of our money into industrial biotech applications in the UK just now, and health applications to some extent. I'm not sure whether we'll succeed spectacularly in either of them, but we're trying.
Okay, thank you very much, everyone. Unless there's like one last burning question, maybe you can grab some speaker on the way out. But I think we should let them go to their homes. We're not keeping anyone here, so thank you very much to everyone coming. I know it's like exams are coming in soon. I have one today. Thanks for speakers for being here with us.
Thanks to you guys for organising it.
Yeah, there weren't for the question.
It's good to have some critics to the session.